On tropical reefs around the world, nothing sees the way you'd expect. The most visually sophisticated animal on the planet isn't a hawk, an eagle, or a deep-sea fish shaped by millions of years of darkness. It's a crustacean the length of a hot dog, hiding in a rubble burrow in shallow water that you could theoretically wade out to in knee-deep water and scoop up with your hands. We're talking about an animal that punches hard enough to shatter aquarium glass and generate a flash of light in the process, whose eyes swivel independently in opposite directions to scan different parts of the reef simultaneously, and whose visual system processes 16 separate channels of color information compared to our 3. According to everything we thought we understood about animal vision, a creature like this had no reason to exist. But here it is. So, how did one small crustacean end up with the most complex eyes ever documented on Earth? What is it actually seeing that no other animal on the reef can? And what happens when scientists spend 40 years trying to understand a visual system so alien to our own that the gap between knowing how it works and experiencing it might be permanent? This is the story of the mantis shrimp. And it's stranger than anything we could have invented. Before we go any further, if you're new here, take a moment to subscribe to this channel for more deep ocean documentaries like this. Now, let's get into it. In a university laboratory in the early 1960s, a marine biologist came in one morning to find his aquarium tank in pieces on the floor. Not cracked, shattered. The glass had been 1/4 of an inch thick. The tank's occupant, a small, armored crustacean barely the length of a ruler, was still inside, rotating its eyes on their stalks to look at whoever had just walked in, apparently unbothered by the carnage it had caused. The researcher did some rough calculations. The force required to produce that result was, by any estimate, impossible from an animal that weighed perhaps 100 grams. He noted the incident in his field log as remarkable strike force, source unknown, and moved on. He had no idea what he was dealing with. And at the time, honestly, almost nobody did. For most of the 20th century, stomatopods, the order of crustaceans to which the mantis shrimp belongs, were a footnote in marine biology. Researchers knew they were colorful and aggressive. They were hard to keep in captivity, harder to study, and nobody was funding serious work on a creature most people had never heard of. So they sat on the shelf, scientifically speaking, for decades. That started to change in the 1970s, when a behavioral ecologist named Roy Caldwell at UC Berkeley began the first real systematic study of mantis shrimp behavior. What Caldwell found over years of fieldwork was an animal nobody had given enough credit to. These things maintained territorial boundaries with a level of precision that implied they could recognize individual rivals. Multiple species formed monogamous pair bonds that lasted for decades, which is frankly remarkable for a crustacean. They had complex signaling behaviors that nobody had bothered to decode yet. These animals, Caldwell reportedly told a colleague in the mid-1980s, are not seeing the reef the way we see it. They're seeing something we can't even imagine. At the time, it sounded like a hunch. It turned out to be a dramatic understatement. The real breakthrough came through a physiologist named Thomas Cronin. Cronin had spent his early career studying the photoreceptors of ordinary crustaceans, crabs, shrimp, reef fish. Unremarkable stuff. He stumbled into stomatopod vision almost by accident, pulled in by a colleague's mention of some anomalous data that didn't fit any existing model. That data consumed the next 4 decades of his professional life. In 1994, Cronin and the visual ecologist Justin Marshall published a paper in Nature that stopped the vision science community cold. The mantis shrimp, they reported, possessed not 2 types of photoreceptor, not 3, not the record-holding 4 found in pigeons, but 12 to 16, depending on the species. Each one tuned to a specific narrow band of wavelengths. Together, spanning a range from deep ultraviolet at around 300 nanometers to far red beyond 720. No animal on Earth had ever been described with a system like this. The paper left one question conspicuously unanswered. To understand what makes the mantis shrimp's eye extraordinary, you first need to understand what your own eye is actually doing, because it's probably not what you think. You have 3 types of color-sensing cells in the back of your eye. Each one is sensitive to a different, broad range of wavelengths, roughly red, green, and blue. When light hits them, each sends a signal to your brain, and your brain compares those 3 signals to work out what color you're looking at. That comparison is doing all the work here. The color you see isn't sitting on the surface of an object waiting to be read. It's calculated. Your brain receives 3 numbers and constructs an answer. It's a clever system. From just 3 receptor types, humans can distinguish around 10 million different colors. That's genuinely impressive. But it only works through comparison. 3 broad, overlapping windows. Brain does the heavy lifting, and the result feels from the inside like direct contact with reality. It isn't. A dog with 2 receptor types operates on a simpler version of the same model, which is why the whole "dogs are colorblind" thing is sort of true. A pigeon with 4 operates on a richer one, able to discriminate colors in the near ultraviolet that we can't see at all. And a mantis shrimp with 16 operates on something that comparison-based models don't even begin to capture. The mantis shrimp's eye is a compound eye, like you'd find on a fly or a bee, but the similarity stops there almost immediately. Running across the center of the eye is a horizontal stripe of specialized optical units called the midband. Think of it as the engine room of the whole visual system. 6 rows of tightly packed structures, each doing something different. The first 4 rows are where the color magic happens. They stack photoreceptor layers on top of each other, separated by biological color filters made of pigment molecules embedded directly in the receptor tissue. These filters narrow the wavelength range each receptor responds to down to windows as tight as 15 to 20 nanometers. That's staggeringly precise. For comparison, one of your cone cells responds to a range of over 100 nanometers. The last 2 rows of the midband handle polarized light. And here's where things get really interesting. Polarized light is light whose waves oscillate in a specific plane rather than all directions at once. Your eyes are completely blind to it. You literally cannot detect it without instruments. The mantis shrimp has 4 separate receptor types dedicated to polarized light, each sensitive to a different orientation. It can distinguish multiple planes of linear polarization and, uniquely among all animals studied so far, circular polarization. That's light whose waves rotate in a helix as they travel. Add it all up, and you get 12 channels for color detection, 4 channels for polarization, plus separate regions handling motion and wide-field spatial vision, all housed in an eye that rotates independently from its partner on a movable stalk, covering nearly 360 degrees. The mantis shrimp isn't just running more channels than we are. It's running a fundamentally different kind of visual operating system. Before we go any deeper into the eye, though, let's properly meet the animal wearing it. At the margin of a coral reef, in water maybe 2 or 3 meters deep, something watches from a burrow in the rubble. The entrance is barely wider than a human fist, and the creature inside, at first glance, might not seem like much. About 15 centimeters long, segmented and armored like a medieval gauntlet. Banded in colors almost too vivid to be natural. Electric green along the carapace, orange at the joints, flashes of red near the legs. Its eyes sit on independent stalks and rotate with an autonomy that, once you notice it, is difficult to stop staring at. Each eye moves separately, scanning different parts of the visual field at once. This is Odontodactylus scyllarus, the peacock mantis shrimp. Mantis shrimp aren't actually shrimp. They belong to Stomatopoda, an order with a fossil record stretching back 400 million years. That's long before the dinosaurs, long before the first flowers, long before most of the life we'd recognize on Earth today. There are around 450 known species, distributed across tropical and subtropical reefs worldwide. Most are solitary and fiercely territorial. Many are monogamous for life, which, again, is a remarkable thing for a crustacean. They split into two camps by hunting strategy. Spearers use barbed appendages to impale soft-bodied prey. Smashers use something else. The smasher's weapon is the dactyl club, a calcified, heavily mineralized appendage folded at the front of the body like a cocked fist. When it fires, it accelerates from rest to impact in 3 milliseconds. Peak velocity, over 23 meters per second. The acceleration involved, roughly 10,400 times the force of gravity, is among the highest ever recorded for any animal movement on Earth. Impact force against a crab shell, more than 500 newtons, from an animal that weighs around 150 grams. But here's the thing. The strike itself is only half the story. At those speeds, the water immediately ahead of the club drops below its own vapor pressure for a fraction of a millisecond. It vaporizes. The resulting cavitation bubble collapses almost instantly, generating a secondary shockwave and, through a process called sonoluminescence, a brief flash of light. The prey gets hit twice in under a millisecond, first by the club, then by the collapse of a vacuum. That's what destroyed that aquarium wall in the 1960s. Materials scientists at UC Riverside have been studying the dactyl club's internal structure since 2012. What they found was hydroxyapatite crystals arranged in a herringbone pattern that redirects crack propagation through the material. Essentially, a biological impact-resistant composite that outperforms most of what we've engineered. The US Department of Defense has since funded research into applying those structural principles to body armor and combat helmets. So that's the mantis shrimp before we've said a single word about its photoreceptors. An animal that's been quietly running one of the most lethal punches in the natural world and one of the most sophisticated visual systems on Earth simultaneously for 400 million years in shallow reef water you could snorkel in on a Tuesday afternoon. Back to the eye. Because 16 types of photoreceptor is only the beginning. The first row of the midband handles deep ultraviolet, wavelengths around 300 to 315 nanometers, completely invisible to human vision. Most reef fish and invertebrates reflect UV light in patterns that no predator other than the mantis shrimp can see. Their UV visible markings are, in effect, a private channel broadcasting information that most of the reef doesn't have the equipment to intercept. For a mantis shrimp hunting in a system full of competitors, that's a significant advantage. Rows 2 through 4 move up through the visible spectrum, each receptor type filtered to a precise 15 to 20 nanometer window. 12 channels total for color, each one a specialist covering its own narrow slice of the spectrum. And then there's the polarization vision, which does something even more specific. Several mantis shrimp species reflect circularly polarized light from structures on their own bodies, patches on the carapace, the appendages, the face. This signal is only detectable by animals with the specific circular polarization receptor needed to receive it. On a reef full of fish, cephalopods, and other crustaceans that can't detect it, this is essentially an encrypted communication system. Mantis shrimp can signal to each other in a code that the rest of the reef is deaf to. Status, aggression, mating readiness, all broadcast on a channel nothing else can even perceive. 16 channels, ultraviolet prey detection, multiple planes of polarization, a private encrypted channel for intra-species communication. You'd be forgiven for assuming this made them the single best color-discriminating animal on the planet. You'd be wrong. And finding out why is where this story gets genuinely strange. In 2010, a research team led by Hannah Thowen at the University of Queensland set out to measure exactly how finely mantis shrimp could tell colors apart. Standard behavioral experiment. Train the animals to pick a specific colored light for a food reward, then test how similar two lights can be before the animal can no longer reliably distinguish them. Humans can tell apart colors separated by as little as 1 to 4 nanometers across much of the visible spectrum. We're genuinely good at this because trichromatic comparison extracts fine color differences from the ratio between 3 broad signals. More channels, you'd assume, means finer discrimination. The Mantis shrimp failed at 25 nanometers. 25. Worse than a honeybee, significantly worse than a human. Within the 15 to 20 nanometer bandwidth of a single receptor type, they were, for all practical purposes, colorblind. More photoreceptors, worse color discrimination. That result should not exist, but it does. What it suggests is that Mantis shrimp aren't doing comparison-based color vision the way we are. They're not blending channels into a composite experience. Each receptor appears to report independently, a parallel bank of narrow band detectors, each firing or not firing based on whether its specific slice of the spectrum is present without checking in with its neighbors. They're not mixing colors, they're reading them. So if 16 channels aren't for fine color discrimination, what are they for? The best explanation the science currently has is called spectral sorting, and it reframes the entire question. The idea, developed by Marshall and Cronin through the 2000s and 2010s, goes like this. Because each receptor covers such a narrow, precisely defined slice of the spectrum, the Mantis shrimp doesn't need to compare channels to identify a color. It just reads which receptor is firing most strongly. If the 450 nanometer channel activates, that's blue. If the 530 nanometer channel activates, that's green. No comparison, no calculation, no blending. Just which channel fired. Think of it less like mixing paint and more like reading a barcode. A scanner doesn't blend colors to figure out what product it's looking at. It reads which lines are present in sequence and outputs a result instantly. The result is categorical rather than continuous, but it's also incredibly fast. On a reef where prey needs to be identified in milliseconds, where rival mantis shrimp display specific color patches as social signals, and where mates advertise fitness through patterns tuned to precise wavelengths, that kind of fast, categorical identification might be worth a lot more than the fine gradation that comparison-based systems provide. Speed over nuance, recognition over discrimination. And the UV and polarization channels add layers to this that have no parallel in our own vision. UV reflective patterns on prey that competitors can't see. Circularly polarized signals shared exclusively between members of the same species. The Mantis shrimp isn't just operating on more channels. It's operating on channels that simply don't exist for most of the ocean around it. It's a convincing hypothesis, well supported by behavioral data, physiology, and evolutionary modeling. But Cronin himself is careful to call it that, a hypothesis. Because the honest answer to what are 16 photoreceptors actually for is, after 40 years of research, still, we're not entirely sure. And that uncertainty is actually the most interesting part of this whole story. In 1974, a philosopher named Thomas Nagel published a short essay called "What Is It Like to Be a Bat?" His argument was deceptively simple. Bats navigate using echolocation, sending out sound pulses and reading the returning echoes as a spatial map of the environment. We can describe this process in extraordinary detail. Frequencies, timing, neural processing, behavioral outputs. We can know everything about the mechanism. But here's the thing. No amount of that description brings you one step closer to knowing what echolocation feels like from the inside. What it is to experience the world as a texture of returning sound. That knowledge isn't unavailable because we haven't looked hard enough. It's unavailable because you'd need to be a bat to receive it. Apply that to the Mantis shrimp. We know the peak sensitivity of each of its 16 receptor types, specified to within a few nanometers. We know the structure of every filter in the midband, the geometry of every photoreceptor layer, the neural pathways from the eye to the brain. We know the behavioral outputs of the system with considerable precision. We've published hundreds of papers. We've built computational models, and we still have no idea what the reef looks like from inside that eye. Cronin said something in a 2020 eye, "Enitama som hari, santi aventure fro, wall and fui, and tro the witche, arca interview dakshki, and diaf, so it's hard to forget. Oh, he'd been asked whether, after 40 years, he felt he truly understood how mantis shrimp see. He paused before answering. "I know a great deal about how the system works," he said. "I have no idea what it's like. 40 years, hundreds of papers, the world's foremost expert on the system. And the honest answer is, I have no idea what it's like." That's not a failure of science. That's science being honest about its own edges. There's a difference between understanding a mechanism and accessing an experience. The mantis shrimp sits on the far side of that boundary, in a visual world that no human mind, however well informed, however precisely equipped, can enter. We can measure it, we can diagram it, we can name every channel. We just can't see it. And here's the part that makes all of this more urgent than it might seem. Mantis shrimp are reef animals. Almost all 450 known species live in the shallow tropical and subtropical reef systems that are currently among the most climate-stressed environments on the planet. Reef bleaching events, triggered by rising sea temperatures, have killed enormous areas of coral across the Indo-Pacific, the Caribbean, and the Red Sea over the past 2 decades. And when the reef structure goes, the complexity that mantis shrimp depend on for their burrows, their hunting grounds, and their social signaling goes with it. But the threat isn't just habitat loss, it's also time. We've formally described around 450 stomatopod species. We have detailed physiological data for a small fraction of those, the visual ecology of the vast majority, which specific receptor types they carry, what wavelengths they're tuned to, what signals they're sending on channels the rest of the reef can't intercept, is almost entirely unknown. We've mapped the visual system of a handful of species and extrapolated, but the mantis shrimp family is 400 million years old, has radiated into hundreds of distinct species across wildly different reef environments, and in all that time has had every reason to evolve different visual solutions for different ecological problems. We could lose those solutions before we even know they exist. Research has shown that rising temperatures combined with the physical degradation of reef habitats could push multiple stomatopod species toward extinction within decades. And unlike more widespread marine animals, many mantis shrimp species are specialists. They're tied to specific reef types, specific depth ranges, specific thermal gradients. The same precision that makes their visual system remarkable is also what makes their populations fragile. They're not built to be flexible. They're built to be exactly right. That's a hard combination when the environment starts changing faster than evolution can keep up. It's been 30 years since Cronin and Marshall's 1994 paper told the world that one small crustacean was running 16 channels of visual information that nobody had predicted and nobody fully understood. In that time, we've made enormous progress. The receptor types are mapped, the filter chemistry is characterized, the polarization system is documented, the spectral sorting hypothesis has real support. New mantis shrimp species are still being described. New visual configurations are still being found. Research is accelerating. And still, the fundamental question, what does the world actually look like through 16 channels, remains open. But maybe that's not the point. Maybe the more important thing the mantis shrimp has given us isn't an answer. It's a question we didn't know to ask. Every animal alive is sealed inside its own perceptual world. The dog navigating by a scent trail left hours ago by animals it's never met. The electric fish sensing the shape of nearby objects through distortions in an electromagnetic field it generates itself. The star-nosed mole processing touch through 25,000 sensors on its face faster than the human brain processes sight. Each one experiencing something real. Each one living inside a version of the world that no other kind of mind can fully share. The mantis shrimp's visual system is the most measurable, most precisely documented example we have of a mind operating on a completely different set of inputs than our own. We can count its channels. We can specify its sensitivities. We can model its outputs. And all of that precise, hard-won knowledge still leaves us standing outside a door we can't open. There are colors being perceived right now on a coral reef in the Indo-Pacific that have no names in any human language. Polarization patterns carrying social information in a code the rest of the reef can't read. Ultraviolet signals invisible to everything except the eye that evolved to receive them. They've been there for 400 million years, long before we had the tools to detect them, long before we even knew to look. We're still learning to look.